Hello, in this video we will discuss about the antibiotics effect in the different region of the bacteria. So the effect of the antibiotic we will discuss. So first of all, the antibiotic means the substance which kill the bacterial cell. So it can kill the bacterial cell by damaging their part of the cell. So let's begin to understand here. If first of all the bacterial cell, which that is contained outer is the cell wall and outermost layer is the slimy, is known as the capsule, while inside is the cell membrane and outer is the cell wall. Cell wall is made up of peptidoglycan and lipoic acid and teichoic acid. So here is the PABA. This PABA is go inside to convert the dihydrofolate into the trihydrofolate, and this is the basically the folic acid metabolism in the bacteria can be inhibited by the trimethoprim and sulfonamide, which that is different is the dihydrofolate inhibition by a sulfonamide, while the trihydrofolate conversion is trimethoprim. This is a folic acid metabolism inhibition, and the folic acid metabolism is very important for the bacteria, so for normal growth. So this will be inhibited through a antibiotics. These two type of antibiotics can be inhibit this process, and in this way the result is that the the bacteria will be damaged and killed. So the here is a DNA gyrase. This DNA gyrase enzyme is used for the supercoiling prevention. So the supercoiling will be occur to damage the DNA. So this is the nalidixic acid and leprofloxacin is the important is the class of the quinolone is basically important for the inhibition of the DNA gyrase activity. While here is the protein synthesis from the ribosome 30S is the smaller subunit will be inhibited by the different type of antibiotics: tetracycline, spectinocycline, streptomycin, and givatamycin and thymycin, as well as the amikacin and nitrofurans. And here important is another is the cell wall synthesis inhibition. So the cell wall synthesis inhibition can be occur through a cycloserine and the vancomycin and bactericin and penicillin and cephalosporin and monobactam and carbapenem. This is the seven type of antibiotics can inhibit the cell wall synthesis. So here is important is the cytoplasmic membrane structure. You can see that will be inhibit the cytoplasmic membrane through a polymyxin and datomycin. So the different antibiotics can get the different target of the bacteria. So here is the protein synthesis from the ribosome 50S can be inhibited by the erythromycin and chloramphenicol and clindamycin and lincomycin, and this is the special inhibitor antibiotics. So here is the RNA elongation inhibited by the actinomycin also. So let's begin to understand another way to understand about the protein synthesis through transfer RNA. This protein synthesis through transfer RNA is can be inhibited by the mupirocin and puromycin. This is the two type of antibiotics can inhibit the transfer RNA, so the synthesis of the protein will be inhibited. In this way, the cell will be damaged of the bacteria. So antibiotic is using is harmful due to our microbiome in our gut will damage which is beneficial mutualism and produce the short chain fatty acid as well as vitamin K, and this antibiotic frequent uses will cause antibiotic resistance due to bacterial modification adaptation. Our immune system will not work, so immune response will be decreased, and again disease chances will rise 100%. And in this way, the supplementary vitamin K and short chain fatty acid should be supplied after the antibiotic injection due to the killing of the beneficial bacteria will be also killed, so that is the side effect. And in this way, the basically the immune response is also decreased due to this effect. So the antibiotic resistance we will discuss first. Here you can see this is the bacteria contain a plasmid. Plasmid is the antibiotic resistant genes containing DNA. This is a super extra smaller circular DNA which that produce the antibiotic resistance protein. This, for example, here is the antibiotic altering enzyme which that convert the antibiotic into the another form, while here is after the conversion. So antibiotic when enter into the bacterial cell, it will be degrade, no, not degrade, but it will be pump out from the efflux pump, and this pump out move to outward, so that's effect of the antibiotic will not so longer. So in this way the bacteria can survive. And on the other hand, here is the you can see that messenger RNA will bind with the ribosome is known as the polysome, and this 50S and 30S Svedberg unit collectively is known as 70S, and this produce synthesis the protein. This synthesis of the protein can be antibiotic can enter inside, and this antibiotic will not prevent the synthesis of the protein due to the resistance. So on the other hand, here is the another antibiotic degrading enzyme which that move out from the cell, and after the move out from the cell will bind with the antibiotics. After the binding with the antibiotics, it will be degrade the antibiotics. So degradation will be occur, and in this way the response of the antibiotic will not so longer, and in this way the cell can be survived due to the antibiotic resistance through a plasmid antibiotic resistant genes producing the antibiotic adaptation resistance adaptation. So in this way you can see that is the adaptation through sexual reproduction. So the sexual reproduction is very important for the adaptation through conjugation, transduction and transformation. So the conjugation means the pillars, sex pillars will combine together to exchange their genetic material through a plasmid, while the transduction mean the third party of the bacteriophage is required for it for the transduction, and the transformation, the total transfer of the DNA into the another cell, which that is the harmless convert into the harmful. So thanks for watching.